Hi everybody, welcome to your meiosis video notes. So this is going to break down each of the stages of meiosis, but also talk about some issues that can happen during meiosis. So we start with just some basics. So meiosis itself is cell division that produces something known as germ cells, or they may also be referred to as gametes, and those are sperm and egg cells. So in this process, one parent produces 4 daughter cells, and there are 2 divisions total, which is different than mitosis. Also important to note, the daughter cells have half the number of chromosomes as the parent cell, and we'll get into diploid, haploid in a couple minutes. So we said they are sex cells or gametes or germ cells. So human gametes have 23 chromosomes, but humans, we have 46 chromosomes in all of our cells. How is this possible? Well, you get 23 chromosomes from the egg that made you and 23 chromosomes from the sperm cell that made you, resulting in 46 chromosomes that occurred via fertilization. So as I said, haploid and diploid. So think haploid or half, which is n number of chromosomes. So eggs and sperm cells are haploid. Whereas body cells, or like a zygote, which is a fertilized egg, those are diploid because they have the full number of body cells. Chromosomes are also pretty cool because they can tell us some other things. So a karyotype is a picture of all the chromosomes that an organism has. And you may notice that they're grouped relatively together by size and shape. So if a chromosome is really long, it's typically grouped with the other one that's the same length, and they're numbered. So this image is a little cropped, but this is a human, so this would have your 23 pairs of chromosomes, resulting in 46. So a karyotype, like I said, shows us how they're paired, but it also shows us gender. So males are XY, females are XX. Also, karyotypes can show us chromosome disorders. So Down syndrome, for example, you have an extra 21st chromosome, like our cute little girl over here, right? Her shirt says, "Am I rocking this extra chromosome?" So she has an extra 21. And then you have Turner syndrome, where you just have one X, so you don't have those 2 X's for a female. And then Klinefelter syndrome is when you're a male, so you have XY, but you have an additional X, which we'll get into genetic diseases later, but it results in some different kind of characteristics. So here's a closer look at those different karyotypes. So we see that this one is a Down syndrome karyotype because you see that there's 3 chromosomes here. And you will have to identify karyotypes and what the issue is if there is an issue. The next one is Turner syndrome, where there's only 1 sex chromosome. So a female should either have 2 X's or there should be an X and a Y. And in this situation, there's only a single X. And then lastly, Klinefelter syndrome. So having a Y automatically makes you male, but sometimes mistakes happen in meiosis and you get 2 X's, but you still have the Y. So you'll notice this here would be someone that has Klinefelter syndrome. So some important things during meiosis to realize is that the DNA only replicates once, which is the same as mitosis. DNA replicates once in S phase and then it doesn't replicate again. However, meiosis is different because the nucleus divides twice. So this is known as reductional division because the nucleus is being reduced with that second division. Also, there's those words again. So the parent cell is diploid and then the resulting cells are haploid. So then why do we even do meiosis? We already did mitosis. We already have lots of cells being made. Why do we need a different process? Well, meiosis creates variation. So if you look at these kittens here, they're all from the same litter, but they all look differently. And more importantly, genetically, they're all different. So if we were walking around all clones of each other, we would all get the same diseases, we would all have the same issues. So being genetically diverse allows better species survival. So that's kind of the whole purpose, is to ensure survival of the species. So when 2 parents reproduce, you get variety because those gametes are randomly combining. Asexual reproduction is just a clone and they're genetically identical, which is what you would get out of mitosis. All right, so now into the divisions. So there are two divisions of meiosis. There's meiosis I, known as reductional division, and meiosis II, known as equational division. So first up, meiosis I. So we start with interphase, and this is the same as mitosis. You have G1, you have S, you have G2, and in this stage, your chromosome condenses, it replicates, your organelles replicate. All of those things happen just like it would in mitosis. Prophase I is also very similar. Your centrioles are going to form your spindle fibers, except one little difference here. Chromosomes are going to find something known as their homologous partner. And review, homologous, so prefix homo is same. So it's the same structured chromosome. They pair together. And remember, chromosomes pair by size and shape. So they find their homologous partner. But something called crossing over can also occur here, which we'll talk about in just a second. So this crossing over is where chromosome segments exchange. So that's like, I don't know, you and your best friend saying, hey, I really like your arm. Do you want my arm? And you swap arms, right? Obviously, that would be very traumatic and not real life, but that's kind of what chromosomes do. So they swap out their arms, and then as a result, they have even more genetic diversity. And here's just a visual to kind of show you. So you see the orange and the red, and then they swap, and then your final here is you get two chromosomes with a little bit extra diversity. Metaphase I is also the same as mitosis, except they're in pairs. So the pairs line up on the equator. It's not single file like it is in mitosis. In anaphase I, those spindle fibers are going to shorten, once again pulling the chromosomes apart. But notice how we are moving entire chromosomes, not chromatids to the poles. So we're just splitting the pairs. In telophase I, that spindle is going to break back down, the chromosomes unravel, you get two diploid daughter cells, and they have the same number of chromosomes as the parent cell. So in humans, if we started with 46, remember it duplicates an S, then it reduces back to 46 and 46 in your two cells at the end of meiosis I. Also, I want you to pay attention to the fact that there are Roman numerals. When you are tested on this, if you don't include Roman numerals, that means you're talking about mitosis. So the Roman numerals mean that it's meiosis, and that designates meiosis I if it's 1, or meiosis II if it's 2. All right, so there's also this weird step in between called interkinesis, and it just means between divisions. So it's essentially a split of interphase, which is first, and cytokinesis, which is last. So those two words combine, you get interkinesis. So nothing happens here. There isn't any DNA replication. It is not like interphase. It's kind of just like a timeout before we continue dividing. So now we're going into meiosis II. So we start with prophase II, and notice we have two cells. So there's two cells going through this. So spindle begins to reform, we recondense into chromosomes. In metaphase II, this looks an awful lot like mitosis. We're lining up single file here, except remember it's two cells. So think the Roman numeral, the 2, means two cells are going through this process. Anaphase II, those spindle fibers are going to shorten and pull them apart into chromatids. So the first anaphase I pulled them into chromosomes. Anaphase II pulls them into chromatids. Telophase II, they're going to reach the poles and fully separate, but remember, cells do not fully pinch off and divide until cytokinesis, which is here. So meiosis II ends with cytokinesis, which results in 4 haploid cells. Key word, right? Haploid, meaning they have half the number of chromosomes. So if this was a human, there would be 23 chromosomes in each of these cells. All right, so now we have to get a little bit more into eggs and sperm, specifically, and how meiosis is different if you're a female or male. So female meiosis is also known as oogenesis, and this only results in one cell that can be used for reproduction. And then you get 3 polar bodies, which kind of just disappear in the body. They're not used for anything. Whereas male meiosis or spermatogenesis results in 4 usable sperm cells. So Males produce, as a result, a lot more gametes that are available for reproduction than females. Also important to note, with females are born with all of their eggs. So if you're a female, when you were born, you had all your eggs you're ever going to have in your body, and then they kind of stay in interphase until you hit puberty. And then you start ovulating, and then you have monthly periods. So this is where the egg production starts to finish out the process of meiosis, is once you hit puberty. Males just create sperm cells once they hit puberty, and they have an unlimited number. It doesn't matter. So fertilization then is when the egg and sperm combine, bringing 23 and 23 together, if you're a human, to make 46 into a zygote, which is an important word to know. That's a fertilized egg. So here's a closer look at male versus female. So the oogenesis is female. So it shows at the top here, you have your regular before birth, after birth. So you have all your eggs you're going to have, and then you don't have anything happen until you hit puberty and you go into that process of those eggs maturing. Spermatogenesis is different. And also notice we have 4 usable cells at the end. If I go back really quick, there's only 1 egg and 3 polar bodies, which kind of just dissolve in the body. So males have 4 usable. And then lastly, back to those potential issues. So we talked about how you can have extra chromosomes, but how does that happen? Well, that term is called non-disjunction. So this is when chromosomes don't separate where they normally should. So disjunction means to separate or discombine, and non-disjunction means not separating. So failure to separate when they're supposed to. So this is how you get your genetic disorders, because you end up with a sperm or an egg cell that has an extra chromosome or it's missing a chromosome. So for example, if we look at this top picture, if this was an egg cell and that was chromosome number 21, so there's an extra one, and that sperm cell fertilized an egg, you would have a baby that had Down syndrome. On another hand, if you had this one, so this is a sperm cell, again, let's say it's a sperm cell that doesn't have a gamete or sex cell in it, so it doesn't have an X or a Y, and it were to fertilize an egg, you would potentially have a child with Turner syndrome or a single X. All right, and that is all for your notes.